I thought what I would do is really kind of tell you a story of kind of how I'm all involved in this, and I think that it really touches on all your questions. I am a professor at heart and a storyteller.
Mm-hmm.
Maybe I can tell you what I think is going, what happened in my career and where we are now, because it led to the starting of the Neglected Global Diseases Initiative at UBC over 10 years ago that I co-founded and was the first director, and now I'm the director again, back 10, 11 years ago. But to get to that point and all the work that we're doing, I have to tell you kind of what happened to me, because I kind of fell into NTDs, almost 25 years ago, sort of by mistake, and then have kind of been involved since then. So I did my PhD work at MD Anderson Cancer Center in Houston in clinical pharmacology, but I actually was involved with the development of a lipid-based drug delivery system, IV, for the drug called amphotericin B. So amphotericin B is a polymacrolide antibiotic that is used in the treatment of bloodborne fungal infections and parasitic infections like visceral leishmaniasis. But when you give it IV in one of the formulations, it's really kidney toxic. And in fact, it's got this nickname called amphoterrible. So it's got dose-dependent kidney toxicity. However, the drug is very effective. There's very little drug resistance. Extremely effective against bloodborne fungal infections. So these are infections in patients that are immune compromised. Patients that have that are diabetic, patients that have cancer, HIV/AIDS patients. We've now seen COVID patients. So patients that are immune compromised will be susceptible to these fungal infections. And then the drug is also extremely effective against parasitic diseases. And one of the ones is visceral leishmaniasis, which is a very serious parasitic disease in the developing world. There's a sandfly that comes along and stings you and releases this parasite that goes into the liver and the spleen, and within months you die. I mean, it's a devastating disease, and the treatments have limitations, but amphotericin B is a wonderful drug and is very effective in treatment. But one of the problems with AMP-B is actually not its treatment, but its accessibility and its parenteral form, and I'll get into that in a couple of minutes. So I was a graduate student. My professor developed a lipid-based formulation of amphotericin B. So what happened is that as a graduate student, we developed this formulation. It's called Ableset. It's on the market.
Mm-hmm.
There's actually a better product now called Ambisome, and it's a lipid-based parenteral IV formulation that's less toxic, and you can be more aggressive to patients. However, it is still an intravenous formulation. So after I did my PhD, I did a postdoc in Cleveland, and I was recruited to UBC, and I was an assistant professor and worked all my way up. So I started at UBC in the early 90s. And in late 1999, I was giving grand rounds at Vancouver General Hospital to infectious disease docs about combination therapy of liposomal AMP-B parenteral with other antifungals to treat really nasty fungal infections.
Mm-hmm.
And a bunch of IV doctors in the back of the room said, Kish, could you develop an oral formulation of amphotericin B instead of a parenteral form? Because then we could give it prophylactically to patients in advance before they have to come in for transplantation. So, for instance, AMP-B was used, so when patients were going to have a lung transplant or a kidney transplant, they would have to be on immune-compromised drugs, so there wouldn't be any organ rejection,
mm-hmm.
but that would open them up to systemic fungal infections.
Mm-hmm.
So they always put them on amphotericin B to prevent the fungal infection. But the problem is they had to bring them into the hospital. And so that was a problem. So they said, could you develop an oral formulation of AMP-B that we could give prophylactically to patients prior to them coming into the hospital? And then, they would be pre-treated and loaded up with it and so on and so forth. And I said, well, I don't think it's possible. I don't think AMP-B is possible. We can absorb it at a high enough concentration, get around the dose-dependent kidney toxicity, so on and so forth. To make a long story short, went back to the lab and created an oral formulation of AMP-B that was effective in our animal models against systemic fungal infections. And then one day, I had an undergraduate medical student come to my office and said, have you thought about treating oral ANT-D to treat leishmaniasis? The big problem is that current treatments were not accessible in the developing world. They were mostly to patients that were women and children, women of childbearing age, children that were out in rural communities that were, working in those rural areas, and they were, because of the poor sanitation conditions, because they were out in the field, they were getting susceptible to VL, to visceral leishmaniasis. The IV formulation was too expensive, was not tropically stable. You had to come into the hospital to get an IV infusion and be monitored for toxicity. So patients were just not getting the IV formulation of ANT-D, and there were also not very many good alternatives. So he said, could you do oral amphotericin B? Maybe that could be an alternative, because then you could take the medication to the people. If you could make it tropically stable, it'd be more accessible, it'll be cost effective. That would be awesome. And I said, I didn't even think about this.
Mm-hmm.
And so we started to work on it, and we caught the eye of the Bill and Melinda Gates Foundation. So the Gates Foundation, I was in Vancouver at the time, had us down to present our work, and they partnered us with the Consortium for Parasitic Drug Development. So this is a group out of North Carolina that the Gates Foundation had funded to develop new therapeutics for Chagas disease, African sleeping sickness, and for leishmaniasis. So they said, hey, this guy up in Canada may have a formulation for VL. So we got partnered by the Gates Foundation with the Consortium for Parasitic Drug Development, and Gates funded our work to develop a new formulation that was tropically stable, because the first formulation, I had it for refrigeration because I wasn't thinking about the developing world.
Mm-hmm.
And then what was really cool is they had these VL and visceral leishmaniasis animal models. And so a third party, my postdoc flew with the formulation to Ohio State University, where they had the animal model that was funded by Gates, the visceral leishmaniasis animal model, and we tested it in their model. And I got to tell you, it blew everybody away. It was like 99% reduction in the VL load within 5 days of our treatment. I mean, we published the paper in Journal of Infectious Disease. Everyone was blown away by this.
Mm-hmm.
And so at that point, we then marketed, or we then licensed the amphotericin B technology to Ico Therapeutics, a local company in Vancouver. But we told them that, there were two major indications here. There is the indication for systemic fungal infections for patients, as I mentioned earlier, that, were transplant patients or HIV/AIDS patients or so on and so forth. But there's this philanthropic aspect, which was to help people in the developing world. We don't want to make money off the backs of those people. It was meant to be, at or below cost.
Mm-hmm.
At that time, UBC developed a global access initiative policy, and we actually published a paper on it. And what the policy was is that certain technologies that UBC developed would be developed at or below cost for developing world indications. And our oral ANT-D became one of the main products in that global access initiative. And that led us to thinking, well, and I got to tell you, I try to get money to do all this kind of research. CIHR wouldn't fund it originally. Eventually they did. I was running all over, and it was tough trying to get research funding for a neglected tropical disease millions of miles away from, from Canada, where you didn't have an infection. So I made a comment that I said, I wonder if there are people on campus at UBC that are having the same issues I'm having.
Mm-hmm.
And I made the joke that byline was, there are neglected people doing neglected disease research. Well, we did an environmental scan. I got some money from our VP research. He said, okay, Kish, here's some money for some summer students. Go and see what's happening at UBC. And oh my God, hundreds of people were doing neglected tropical disease research, and we're under the radar.
Mm-hmm.
So we started the Neglected Global Diseases Initiative in 2010. And what this organization was, it was supposed to be a facilitator. It was supposed to be kind of the champion for these people, to bring their work and share it with the world, help them get funding, PR their work. And a major component of it was actually students, and so forth. So that started NGDI. NGDI was started by myself and Dr. Bob Hancock, who's a microbiologist, and I was the first director. And for 4 years, and you can go to our website, www.ntdi.ubc.ca, and you can see all the folks on there and all the projects. And we raised about $50 million to help with NGDI projects over the last 10 years or so. And that's how NGDI got started. So it became more than just my work with oral amphotericin B, which now has finished phase 1 clinical trials, the safety studies, and we're about to go into phase 2 clinical trials, and it's been delayed because of the COVID pandemic, but we're hoping to do clinical studies down the road. Yeah. But this became more than that. We now had people from all over campus, and not just in the basic sciences and the medical sciences. We had engineering students. We had people in the history department. We had people in the economics department. We had public health people who all wanted to be involved in all different aspects of neglected global diseases. And so it really became a beacon. And so I ran it for the first 4 years, and then I ran off and became a dean from 2014 to 2019, and I turned the leadership over to my colleague, Dr. Rich Lester, at NTDI UBC. And then after I finished my deanship and I finished 1 year administrative leave, Rich asked me, would you like to get back involved? And in fact, we just had our board meeting last week where we just rebranded ourselves just a little bit. We're going to focus on antimicrobial resistance, do some COVID work, vaccine work, sepsis work. So we're refocusing ourselves a little bit. But we're still around, and we've got support from the university. Our president's big into global health. And so not only am I involved in global health with the technology that we developed in my lab that I'm still wanting to help people with VL. And obviously we're going to try to get through a phase 2 trial and so on and so forth. But I've got to tell you that it's broader than that, because I'm trying to help my colleagues. Back in 2010, I was asked to be on the scientific advisory board for Peter Singer's Grand Challenges Canada. And so I was on that board for 5 years and helped review grants and helped with their Grand Challenges program. And of course, a few years ago, got asked by Charles Larson if I wanted to get involved with CNNT and TD, and Allison has been fantastic. And so I'm now on the group there. So my efforts around NTDs, I think, are not only my work with amphotericin B, but much more with the NGDI. Our countries, we span many countries. Most of the work I like to do is, of course, where VL is, so in India, in Africa, in South America. But the NGDI is doing work in Ethiopia, it's doing work in Malawi, it's doing work in many countries around the world. And our partnerships, well, we at Gates Money, we have people who've worked with DNDI, we got people who work with WHO. There's a lot of collaborations that are going on between my work and so on and so forth. And I think the thing that we're most proud of is we've shone a light on NTDs in Canada. And I think the reason why it's important to Canada, I would make the following comment. It's important on two levels. It's important on a geopolitical world level, in that, Lester Pearson won the Nobel Prize in the for his work in the late 1950s for the thing he did with the Suez Canal, our former Prime Minister. Yeah. And people forget that. It's because Canada always was a beacon to help the world. We're always the country that, is out there to help the world. So we need to maintain that kind of world reputation. And doing work in neglected tropical diseases is one of the ways we can do that. So I build on the spirit of Lester Pearson. Pierre Trudeau was like that. He was an international man. I just think that Canada has an international reputation for being a peacekeeper, for helping the world. I know when I've gone around the world, my family comes from India. When they say the word of Canada, they say, "Oh, Canada, those are good people. They're nice to everyone. They try to help people." Yeah. So I think from that point of view, I think it's really important that we play a leadership role in these neglected tropical diseases, and we should invest more there. But there's another reason why this is extremely important, and that is our world is shrinking, and a great example is what's happening with COVID. Our world has shrunk because of transportation and climate change. These neglected diseases could end up on our shores. I'm going to give you a great example. There are VL cases in New Orleans, in Louisiana, because of the climate changes down there. After Katrina in 2005, I went down to give a talk, and I had colleagues that were telling me about the VL cases in New Orleans. I'm like, are you kidding me? Yeah. But it is. And because of the travel around the world, and people are coming from all parts of the world to our country, that's partly what has happened with COVID. We cannot be isolationists anymore. So today, the neglected tropical diseases that we work with, many of them may not hit our shores. But you just don't know. Who would have a year ago predicted that, SARS-CoV-2 would be mostly devastating in the United States? And Canada, by the way, is ranked in the top 20 per capita countries with infection. If you go to worldcoronavirusmeters.com, we're in the top, 24th or 25th. So that's why I think it's important to Canada. And as for students, I think students are extremely important. So at NGDI, we used it as a beacon to get students involved in summer student projects, in graduate student projects, in advocacy. We had student groups like UAEM that, students could get involved in. They are the next generation of researchers in this area, and the best way to get involved and get engaged is early on. So my advice for students and young professionals, get involved early. Get involved in groups. Be a summer student. I got into this job by mistake and later in my career, I only started getting really involved after I got tenure, and maybe that's a bad thing because I was so concerned about just getting tenure and so on. And now it's easier because, now I'm close to retirement, so for me it's easier. But when you're a young assistant professor and, you have to publish or perish and so on, and NTDs doesn't work that way, it was difficult to get involved. I regret that, actually. I should have been involved a lot sooner. Yeah. The last 25 years of doing this work has been extremely enriching for me. But the reason why young people and young professionals have a hard time getting involved is because, metrics to be able to measure success in their careers, NTDs don't lend to that sometimes. It's publication records, grants, papers, all that kind of stuff. That doesn't necessarily happen with NTDs. And that's changed a little bit now. I mean, now academics and deans and so on. I was a dean, and I had a bunch of people who were doing, food security work in Ethiopia, and they're making a huge impact with their Pulses work in Ethiopia. And I told them to document it that way, and I would support them when they came up for merit and so on. They didn't publish a paper, they didn't get into Nature, but they were doing incredibly impactful work. So that is why I think that, younger professionals had a hard time getting involved. But I think those things are changing. But my advice to young students and young professionals is get involved early. Get into a student chapter. Get into a group. Work for a professor. Get involved on a national level or an international level. There are so many chat rooms and work groups and so on. Start early because it's really enriching. And I think the best part is I'm thinking about all the former students that I worked with at NGDI between 2000 and 2014. And many of them have gone on to amazing careers. I still share with some of them on my LinkedIn page. And I remember them fondly from my days that they were students in NGDI. Some have gone on to become amazing lawyers. Some have become, global access advocates. Some work for global affairs. One actually works at DNDI. One works for national UAEM. I don't like to benefit off the detriment of others, but I got to tell you something. I think this COVID pandemic has woken up the world to realize that, there's a lot going on in a lot of these emerging infectious diseases. And I know NTDs are not just infectious diseases, but this is a problem. And I think the world needs to wake up and realize that, something that you and I have known for years, that's the world is now realizing that, hey, what about all these other diseases? Oh, what about all this other stuff going on? So it's a big deal. It is a big deal. So it was a pleasure.